endured that ancient cross. How precious is my Savior's blood. Your beauty of heaven wrapped in my shame. The image of love upon death's frame. If having my heart was worth the pain, what joy could you see beyond the grave? If love found my soul worth dying for, how wonderful, how glorious, my Savior's scars, victorious, my chains are gone, my debt is paid, from death to life, and grace to grace. If heaven now owns that vacant tomb, how great is the hope that lives in you. The passion that tore through hell like gold. The
Partly, people treat water that they get from the kiosk and the piped water. So we went further and looked at the proportion of people that say they do chlorination. Because we know chlorination is an effective way of water treatment if it's done correctly. But out of the people that say they do chlorination, we had only 14% of the people who treated like the surface water and 5% that treated the piped water that had the required chlorine levels according to the WHO standards. So the others had said they did chlorination, but the residual chlorine levels were very low that cannot offer any protection to the water. So still, the treatment methods are not effective. So in this slide, we look at the risk factors for contamination of stored water, and these are the factors that we collected through the household questionnaires. So I must say that this is data that is still under analysis, it's not the final thing. And a quick through of this, we found that animal ownership was a risk factor for contamination of water. And the people who own poultry, the people who had poultry and goats, their contamination levels were higher. Why? From what Roberta explained, we know that poultry are notorious and they're all over the place, jumping all over, and even the goats are notorious, so there are higher chances that they access the water sources, the stored water. So container accessed by animals, so we asked whether is the container you store water accessed by animals, and households who had containers that can be accessed by animals had higher contamination levels. Container at the waist height. So the people who had their containers below the waist height had higher contamination levels, mostly because of easy access by animals and even children. You can find your child playing or bathing in your drinking water. Another thing is where the dipper is placed after drawing water. So we also asked, when you draw water for drinking, the cup, the dipper that you use, where do you place it after that? So there are people who place it back into the water, others place it back on top of the container, but others keep it elsewhere. So the group that place their dipper elsewhere has had higher contamination levels, mainly because maybe you do not know where they're going to keep it, and maybe when they go and get it to get another water, it comes with contamination from wherever it was. So we also have this, will definitely share this out. So just a summary of our findings. We realized that sources of drinking water vary with seasons, and rainwater is predominantly used during the wet season. So some of the water sources were more contaminated compared to water that is stored at the household level, although not always. So some of the rainwater and surface water were highly contaminated, and again some of the rainwater and piped water was safe with 0 E. coli counts. We had very few boreholes in our study area. There were very few, and all of them had very low contamination levels. And we say that physical appearance of water should not be relied on when making a decision on whether to treat water or not. And
people, is it animals, where is it coming from? But the problem with many of the techniques used to do this is they involve a lot of genetics, and they're so expensive, well actually they're almost as expensive as solving the problem in the first place. So how can you come up with a technique that's not amazingly expensive and tells you where the contamination is coming from? So that's the basic idea. I'm going to try and explain this fairly complicated microbiology to you now. So probably most people will have seen out there in nature that there's predators and then there's prey. And what happens when you're wandering around your environment is you maybe see one of these, and then it realizes that it spots its prey, and then recognizing this as food because they're from the same place, this bird realizes that this thing is food, so it attacks it and uses it as food. So we've got a predator and then we've got some prey. So I think my colleagues will hopefully support me on this, but if we were looking now down at a test tube and imagining the same sort of thing, there are of course many different types of germs, if you like, microorganisms in there. There's viruses and bacteria. Now you can imagine that, you know, if we zoomed in enough, actually again we might see some things that are predators, and we might see some things that are prey. So over here the prey are bacteria, and over here the predator in this case is a virus, a type of virus called a phage. So the same thing, where these things have grown up in the same environment, when one of these sees one of these, then it will attack it. So the idea behind all the science that's been done by Brighton and to some extent by colleagues from Maseno as well, what happens is there's almost a two-step process, and in the first step, very difficult step, what we try and do is find and grow a plate of a species, if you like, a particular strain of bacteria or the prey that you only find in lots of cattle, or you only find in nature in lots of humans. So isolate it, put it in a dish, and then the second step, add some water to it from wherever in the environment. Then wait and see if these get attacked by something in the water. And then if they do, then you might think, do you know what? That water sample's got predators that came in it from the same place as where these came from. So that's basically what's going on. So I hope everyone's with me. It took me absolutely ages to understand that. So one of the big things about people working on this is there's actually a really promising species of bacteria prey that lots of people have been working on internationally that's found in humans, like a strain of bacteria. It's been tested all around the world, and people have been working on it in places like the UK, Spain, Japan, Sweden. One of the things you'll notice about that list, apart from the very last country on the list, none of those countries have big waterborne disease problems. So one of the things about One Health Water is it means this science can be taken across into, at least start to apply it in Africa, where actually people do have real problems with waterborne disease, as you know. And on top of that, there was a bit of a chance maybe to do a hunt for some local bacteria prey as well, from cattle here in Kenya. And of course, I think it sounded like there was some very promising interaction with Maseno, and also all the work was happening between Oscar is near somewhere, I hope. A lot of background microbiology going on in the Kenya lab. So that's kind of what was happening. And then just really quickly to give you an idea, there is Diogo. I think that's Diogo there, is it? And then this is the really tough work of trying to track down if you like, the human prey. And then over here is some even more tough work, trying to track down the cattle-based prey. And then after that, a lot of hard work starts in wherever Oscar is. His home territory is now up on the screen, I think. And then we've got the hard work of trying to track down these prey, if you like, that might hopefully begin to tell us where this contamination is coming from. So a lot of lab work going on there. And then there's an awful lot of hard science that, to be honest, I don't understand very well, about trying to work out which ones of these you only find in, say, cattle but not humans, and you find lots of them to pull out the most promising types of bacteria. And then eventually, after lots of work, you can pour an environmental sample onto a big tray of isolated bacteria, and if you're lucky, you'll see some predators making little holes in it and starting to attack the bacteria. So that's the kind of idea of what's been going on. Along the way, unfortunately I wasn't here, but I understand, thanks to Dr. Scholastica and Maseno, I understand there was what sounded like a very successful workshop last week with people working on related techniques around this area, particularly around viruses. And so really, that work is trying to figure out how you can work out where contamination is coming from. So, if I was to sort of try and sort of summarise things, you can kind of see, if you think of that idea of predator and prey, and then having a prey that when you see it being attacked, it comes from a particular place, like a cattle or a human. The most promising prey species, the project's given a chance to take some stuff that's been going on internationally, but actually bring it to Africa. And also maybe to do a little bit of work trying to develop similar sort of prey here in Kenya as well, that are more local. But this is still a work in progress, and I think it needs an awful lot more collaboration to bring it forward. Not necessarily with me, but with my colleagues at Brighton. But the key thing is, if we can get this to work, then it would be cheaper than all of these genetic alternatives to try to do the same thing. So I hope that's a reasonable summary, and it took me a very long time to try and come up with the idea of the book. Somebody said, try and make sure your late mother can understand the talk.
Thank you very much, Jim, for simplifying my resource tracking into a hook and a rat.
So this is the time we want to open it up so that we can get a feedback from all of us who are listening. I remember initially when Jim did the introductory bit of it, we posed certain questions. Do we remember them? We posed some 3 questions. Do we remember them? No, we don't. Okay. So we'll be guided by those questions, but we want to open it up so that we get the feedback, we get what you feel about the presentations that have gone on and what your input can be. I don't know whether I want to go presentation by presentation or we just open up. Remember, Prof was asking for quick questions if there were in each presentation, but now this is a time to giving your feedback. And as you ask a question, as you make a contribution, you may be specific to a given presentation, maybe the last one or the first one, or if it's cutting across, then we'll very much appreciate. Okay. Patterns of the data look right. There was something like that. Do you remember? Do the patterns of the data look right? When Mr. Okoto presented the maps, are they a true reflection of what is on the ground? Or certain things were missed? When we talk about the main risks, are they the right ones or not? Those are the kind of feedback we want as far as that first question is concerned, and any other that is present, that you have. Do the patterns of the data look right? And how do we interpret the patterns? That was the second question. How do we interpret the patterns? And then the last one is action. What can be done to make water safer, prevent disease, spread from livestock to people, and reduce diarrhea. But I'm not limiting the session to those. But those ones could be our starting points and any other. So I want to open it up. Can I see those who want to make contribution? I know the first person talking fast in Malaysia. Who wants to be the courageous one to talk fast? Yes.
Okay, thank you very much for this opportunity. I would like to open it up with a form of a question, and this one is going to our presenter, Joseph Okoto. When you're talking about some indicators of risks at household level, one of the findings was that no residual chlorine was 100% risk at household level. My question is, how do you measure no residual chlorine at household level? How do you know that this water has no residual chlorine as a consumer of that particular water? Thank you. Could be we'll add more as we progress.
We want to take maybe 2 or 3 and then we have responses. So the issue of residual chlorine, scholastic.
I would also like to ask a question to Mr. Okoto concerning borehole water. You say that, and we also know that what do we call it? The borehole, in the toilets. So the toilets should be on the lower ground than the borehole. Now my question was, I'm asking on behalf of someone, I know somebody whose borehole is the rivers. So I'm asking that, what if the distance, the length of the toilet is deeper, as much as it is on the higher ground, than the level at which you got your water from at the borehole? can that water be safe? And then, how far, the distance? So forgive my ignorance, but I'm asking how far is the distance between that level, when your water or your borehole is, maybe the distance between the toilet and your borehole. If at all, your borehole is at the lower ground than your toilet. So the distance also matters. How far should it be for the water to be safe? The last one, we go to Dr. Emma. It has appeared that borehole water was the least contaminated water. So how can we make this water softer? Because most people don't prefer this borehole water for use because it is hard water. So is there a good way of making this water softer? Is it chlorination or any other best method? Finally, what would be the best method of treating rainwater, which has appeared to be popular? What would be the best method for treating this water safer for drinking to reduce the amount of contamination? So the safest method of treating this rainwater. Thank you. So that is Dr. Emma also.
Okay. Two more hands and then we can pause for answers before we take the next one, okay?
Thank you. I think these are very interesting and nice presentations. Just as one suggestion, Joseph, you are going away, but I am suggesting that for us to better understand the levels of risk involved in this thing, it might be nice to summarize all the risks together and let us see the percentage risk that will occur if you use water from different sources. I think what you need is that you provide a risk by every item. But suppose, and you say that 10% or 50%, 9%, what would be the risk by percentage, so that all of us know it, so that it looks like a generalized information. The other issue that I want, that struck me very much is the issue that there seems to be some, at one stage in your data, where the risk is low at the water source. Is that right? And then when you take that same water home, the risk is higher. I guess I follow you. First of all, it was not clear to me what was happening on transit between the source and home. And perhaps an intervention would be very useful there. And secondly, what is the difference between the risks that we can see? Is it 50%, 100%, so that it is worth being concerned about? Again, your summary of this tracking is interesting. I wish you could have simplified it a little bit better. I saw the bag about the same vision and the food at its back. And the microbacterial issues are very complicated. And maybe you can give us just a small statement of what happens at the bacterial level.
We'll take this last question and then give an opportunity for response before we take the next one.
Okay, thank you. And my question goes to Dr. Emma. You think that tap water is not safe when you use it directly? Then we believe that it is treated at the point. does it mean that the treatment is not well done at the point, or it is contaminated on the way before it reaches at the point of collection? Thank you.
Thank you. I want to give the opportunity for response so that we can have answers before we move and take the next set of questions.
This residual chlorine, how to know the amount of residual chlorine in water? Actually, chlorine normally is responsible for the compounds like nitrates, the bacteria, other organic compounds. Normally, even the chlorine which is in the market, normally there is an instruction on how many drops. So the small cup is responsible for what amount of water. Normally it is 20 liters for that small white cup. But we actually find people using the small one for even 200 liters or even 50 liters. Now in that case, such kind of water might not have a residual chlorine. This residual chlorine, actually it is meant to take care of any contaminant, if at all, can come into the same water system. Am I clear?
How do you test for it? You are not clear yet. How do you test for that? What method?
We have test strips. Normally it is not at the village level. I have no idea whether commercially it has been taken to the village, but normally the water treatment departments, they do spot checks at each and every water point.
That's what I wanted from you. We take water from the household, okay, and then take water from the source. Now at the household level, if it was not from a formal water supply system, unless it is treated or the person tells us it's treated with chlorine-based detergents, then we test it there and then. So how do we test it? We have chlorine strips, which gives us the quantity of chlorine that is inside that water. We dip it, and then after dipping it, you observe it, and there are guidelines on how to observe it and estimate the amount which is inside. Okay? That's clear. Now the second question was the length of the pit latrine is deeper than the borehole. A pit latrine can never be deeper than a borehole. You know, boreholes go up to how many meters? Some are even 100 meters. Some even 100 meters. There's always a confusion between boreholes and wells. Yeah? A borehole is a type of a well which is very deep. Yeah? But we have hand-dug wells. Okay. We also have machine-drilled wells, and then we have a borehole. A borehole is dug. How to differentiate them? A borehole is dug using a drilling machine, and it goes very low, down to almost 80, 100 or so millimeters. Okay? So I guess that what you meant was that a scenario where you have this is your pit latrine, isn't it? This is your pit latrine, and this is your fecal matter unit. And then it was up or down. The latrine is upstream. The latrine is here. Let us assume that is upstream, and it is deeper than the well, isn't it? Now, this is very serious if it is upstream. Because you know that This is the ground level, isn't it? Let us sorry, because your physics cannot go above ground level. Sorry about that. Let us say this is the ground level. And under here, we have what we call soil matrix, okay? Now, when it rains upstream, or as it rains, yeah, the soil acts as a sponge. Yeah? You are pouring water on a sponge. You are pouring water on a sponge. So when you pour water on a sponge, what happens? It disappears into the sponge because it goes down. So the soil has, a structure that looks like a sponge. There are small pores that when it is raining, it goes down there. But as it goes, these pores are interconnected. They are all interconnected.